Thanks, Carmen, for inviting me, and thanks, Carmen, for really drawing me into the research of ME/CFS and post-infectious syndromes. By now, you should all be convinced that at least in some of the patients, there's autoreactivity. Yeah, so this you should have now kind of gotten with the current session. And I will not talk about autoreactive B cells today because I still don't have proof with the findings we have that they really are autoreactive. But what I will touch upon is the changes, the phenotypes of altered or activated T and B cells we see in patients with ME/CFS. And if autoreactivity plays a role, one can postulate these hypotheses. That some of the patients, not all, and we all heard about the heterogeneity, are characterized by activation of autoreactive B cells and signs for T and B cell interaction, and that the degree of this activation can be utilized for treatment response stratification. And this is all what we are searching for, yeah? To identify before initiating immune modulating treatment, those who will respond. And that is, of course, what I'm very interested in. We will, by elucidating or characterizing the phenotype and the molecular signals of these activated T and B cells, maybe identify novel therapeutic targets. And this is what I set out when we got the funding for a BMBF project, or Carmen got it, and I had the task to coordinate the biomarker analysis. If one wants to study the T and B cell interaction, how it is altered in those patients, we really have to recapitulate what's happening in the secondary lymphoid organs again. So the majority of situations where B cells differentiate, not all, but the majority, where they differentiate into autoantibody or antibody producing cells as a whole, is relying on T cell help. So what's happening is that the T cells and the B cells, and then here, then of course the T follicular helps, they come together at the border between the T cell and the B cell zone. And they do that in a chemokine-driven manner by CXCL13, and therefore need to express, and this is important, the chemokine receptor CXCR5. And then they interact, and they utilize different receptors, ligand-receptor pairs, and I've listed a few of them, and also the T cells help them with producing cytokines. At this stage, they have, in principle, the B cells, they can make 2 decisions, or maybe 3. But in the aspect of autoantibody producing B cells, they can immediately differentiate into autoantibody producing plasmablasts, short-lived plasmablasts, in what we call extrafollicular driven pathway, or they can enter into the germinal center B cell reaction and there receive more help and start somatic hypermutation so that the antibody the sequences will change. And why is that important? Because the way the interaction is happening is different at these 2 different sites. And by elucidating the phenotype of the T and B cells, which sometimes, although it's always better to look into the secondary lymphoid organs, but sometimes they enter the blood circulation. And by looking at the phenotype, you can guess, is there a process happening which leads more to short-lived plasmablasts, or is there a process operational which drives germinal center reactions? Yeah, it helps you a lot because that also helps to define therapeutic targets. And some of the molecules which are involved here, like OX40, CD40 ligand, they are shedded. They belong to the TNF receptor family. They are shedded from the surface of the cells and enter the circulation as soluble molecules. So by measuring them in the plasma, you get an idea on whether they are maybe also expressed in the secondary lymphoid organs. This is what I'd like you to recapitulate if we now enter into the characterization of the T and B cells. The way we do it, and we don't only do it with ME/CFS, is always in a three-step process. We start, and I think this is very important, with a large-scale immune cell phenotyping, where we run multiplex cytometry, and you can either do mass cytometry or spectral flow, it doesn't matter. But it's very important because it has much more power. You study more cells than if you immediately enter into single-cell RNA-seq analysis. And why is that important is that this guides you to which cell types you need to enrich for the then subsequent single-cell RNA-seq profiling, which gives you the molecular targets. And you can also add plasma proteomics, as I just mentioned, because it's indicative of the underlying process. And at the end, with all the information you get, you can then design 2D and 3D co-cultures and run perturbations, which give you a little bit of a mechanistic information. This is how we do it. But now, before entering into ME/CFS, I'd like to tell you a story from a well-accepted autoimmune disease, which is anti-neutrophil cytoplasmic antibody-associated vasculitis. Because we can learn from other accepted autoimmune diseases always a lot. This is an autoimmune disease. The name says it already. You have two main autoantibodies which are binding to neutrophil or myeloid cell proteins, PR3 or MPO. And the main manifestations are in the kidney, but you have also additional organ involvement. And the way the organs are involved varies a little bit between the PR3 and the MPO-driven process. What's happening is that it's depicted down below that the autoantibodies bind to the red dots there on the surface of the neutrophils, and thereby the neutrophils get activated and damage endothelial cells at the bottom. This is in principle. And in both situations, PR3 positive and PO positive, you have kidney manifestations. So maybe it should be very similar. And now we entered into B-cell profiling running this multiplex immune cytometry. On the left side, you see then a UMAP, which shows the clustering of the B cells into the meta B cell populations, naive B cells, memory B cells, plasma cells, and so on. What you see in the middle is how the cells distribute from controls, MPO positive autoimmune patients, and PR3 positive autoimmune patients on this UMAP. It's not published, so please don't distribute it. I just want to use it as a teaser. What you see is the patients differ in their profile from the controls. But what's more important is that the MPO and the PR3 positive patients differ dramatically. You see the B cell activation profile or the process is probably not the same. And you can really follow that if you study the markers involved. And basically, what's different is that the MPO positive ones, they show a very strange CCR6 positive B cell population, whereas the PR3 positive ones have more the classical plasma cell activation. Why I'm showing that is that although everyone assumes this is one disease, the autoantibody profile not only maybe is linked to certain differences in the clinical response, it's also linked to differences in the B cell activation. And this is important if we want to develop therapeutic targets. We see something very similar on the T cell side, where the PR3 positive ones are the ones which have elevated T follicular helper T cell responses. So now coming to the ME/CFS patients. And I was asked by Carmen Scheibenpong, who ran this clinical trial, or my group, not I myself did not pipette anything, but our team had the task to profile patients undergoing immunoabsorption. And this is not a randomized trial, but I will come to one at the end. 20 patients diagnosed with ME/CFS and being positive for autoantibodies. So you already enrich for patients being positive for autoantibodies. And we run the following comparisons. We first wanted to know how the patients differ from healthy controls, how responders versus non-responders differ at the prior to the immunoabsorption, and whether the immunoabsorption can normalize the B-cell profile at the end of the cycles. And here, glimpse onto the B-cell profile, again, UMAP dividing the B-cells into the main populations, memory B-cells, naive, and so on. What you can already also see at the bottom of the UMAP is that the post-COVID patients with ME/CFS differ from the controls, but what you then see is that they have elevated frequencies of B-cells, which are highly positive for CXCR5, germinal center reaction, and are activated with strong upregulation of CD25. So we are talking about a normal germinal center B-cell reaction here. What's important is that it's the responders which show this elevation. It's you can nicely distinguish them from the non-responders prior to the treatment initiation, and you get a reduction of the B cells through the immunoabsorption, which gives you a hint that they may play a pathological role. And this is linked with also elevated plasma blast levels. What one now would need to do, and what we are currently doing, is to link the more early B-cell activation, so these activated naive-like B-cells, to the plasmablast by studying the B-cell receptor sequences and look for clonal overlap. And we don't only have alterations in the B-cells, we also see it in the CD4-positive T-cells. We see in the ME/CFS patients elevated frequencies of activated T follicular helper cells. Here, the levels do not distinguish responders from non-responders. This is limited to the B-cell side. We added plasma proteomics running the NULISA platform from Alamo on plasma samples from the patients prior to the immunoabsorption, and then also comparing again pre and post samples. And what one sees that the patients have elevated concentrations of markers indicative for T-B cell interaction, CD40 ligand, OX40, and that gets reduced upon immunoabsorption. Again, a link that this might drive the pathology. Now, this was a trial on 20 patients only, not randomized. How does it look in a randomized placebo-controlled immunoabsorption trial? This is the trial run by the team from Harald Prüß, and I have to acknowledge many more physicians here and people involved, Hanna Pressler, Federico Ufer, and others. What one can see again when looking at the UMAP, that is that the patients prior to the immunoabsorption have accumulate the B cells at a different site. On the right side there, in comparison to the controls in the area of the memory B cells, and the phenotype is very similar to the one we see with common patients. And it's a placebo-controlled trial. We still don't know who is responding, but what one can see is after treatment that a proportion of the patients, and it's probably the patients responding, normalize their B-cell phenotype again and accumulate in areas where the controls are present. I hope I made up some time or did not extend it too much, but what I wanted to show you is that even well-accepted known autoimmune diseases are characterized by heterogeneous T and B cell responses and phenotypes. So this is something very common, and so we should not expect to see one phenotype in our autoimmune patients. Yeah? It will be very heterogeneous. Then some patients show signs of T follicular helper and B cell activation, which is indicative for an ongoing follicular interaction, and that they might be used for patient stratification, at least the B cells. The plasma proteome data support that, and we are currently performing ongoing work to study the B cell receptor sequences to perform this overlap analysis between the early activated B cells and the plasmablasts, and maybe identify autoantigens. And already the plasma proteome gave us some hints for new molecular targets, but definitely the single-cell RNA-seq, which Anna will then elaborate further, will provide more ideas for novel targets. With this, I'd like to thank my group, all the teams involved here, the patients, of course, the funders, and thank you for the attention with a nice cartoon we always use for our public outreach to teach the school children that damage to one organ, and it might be a respiratory infection, can lead to damage in other organs. Thank you.
Thank you very much. We are running out of time, and remember that we have get together. But still, we have here a very important question from an immunologist in the audience outside. He said that he read in Google on you that you are an expert on T regulatory cells. Would you like to say one sentence, what happened with T regulatory cells in the post-COVID syndrome?
Of course, if you study either in the acute phase, but also in the post-infectious syndrome patients' regulatory T cells, you will even see elevated numbers. Yeah? And this is very established for other autoimmune diseases, because this is a normal counter-regulatory mechanism. So one should be very careful when drawing conclusions for changes in numbers or percentages of regulatory T cells. I think it's all about the balance and whether these cells are able to actually control the ongoing reaction. And so this would be my answer.
Thank you very much. Thank you.